everyone. Welcome to another RRR series of January 2025. And in this series, we will be discussing some important recently appeared questions of the topic on biochemistry. So let's see in 2024, what are the questions we had? So this is the first question. Which of the following metabolic pathway is responsible for the removal of two carbon atoms from acetyl-CoA as carbon dioxide? So here are the options. So they have asked which metabolic pathway responsible for removal of two carbon atoms. So glycolysis, Curie cycle, citric acid cycle, also called Krebs cycle, ETC, or pentose phosphate pathway. The answer of all these things, so you have to select the best option. So the answer is citric acid cycle, also called as Krebs cycle. Okay. And where does it happen? It happens inside the mitochondria. So as you can see, the acetyl-CoA, okay, combines with the oxaloacetate to form the citric acid. Then series of reactions will happen at the junction where isocitrate dehydrogenase, okay, is acting on oxalosuccinate. It is giving the product alpha ketoglutarate. You can see one carbon dioxide is released. Then this alpha ketoglutarate will be converted to succinyl-CoA. Another carbon dioxide molecule is released. So this enzyme is alpha ketoglutarate dehydrogenase. So now it continues to further give back the oxaloacetate. So the question here was, which cycle will release the carbon dioxide from acetyl-CoA? So the answer is citric acid cycle. Let us take the next question. Name the two enzymes which remove the carbon atom of acetyl-CoA as carbon dioxide in citric acid cycle. Now this is a bit twisted question on the same topic. So you got to identify which are the two enzymes. So isocitrate dehydrogenase or alpha ketoglutarate dehydrogenase, aconitase, isocitrate dehydrogenase, aconitase, alpha ketoglutarate dehydrogenase, alpha ketoglutarate dehydrogenase, and succinate thiokinase, or the option number C. So if you carefully observe, all these enzymes are the enzymes of citric acid cycle, which is already mentioned. So you have to identify which of the one is causing that release of carbon dioxide. So the options here is alpha ketoglutarate dehydrogenase and the succinate alpha ketoglutarate dehydrogenase and the isocitrate dehydrogenase. So isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. So which is the option? Option number A, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. So that's the second question. Now we will see another question of 2024. In the double helix, adenine forms hydrogen bonds with which of the following base? Is it thymine, cytosine, guanine, adenine, or uracil? They're asking, it is the double helix, which you have to be careful. So adenine is a purine. So with which other base it will make a base pairing? So the answer is thymine. So what is that pairing? It is called as Chargaff's rule, where the A pairs with the T, adenine pairs with the T, the double bonds are there, and then the guanine pairs up with the cytosine. So in DNA, nitrogenous bases, that is adenine, pairs up with thymine through two hydrogen bonds. So this is a Chargaff rule, okay, which is happening in the DNA double helix, and this will give the stability to the double helical structure. Next question. The process which is responsible for the production of mRNA in cell called as translation, transcription, replication, reverse transcription, or splicing. So here it is production of mRNA from, it is DNA. So the answer is transcription. Transcription is a process where the mRNA is synthesized from DNA template. During this process, what are the enzyme required? RNA polymerase is required. A complementary mRNA strand is formed. Then it is carrying the genetic information for protein synthesis. So replication means what? DNA to DNA. Transcription is DNA to RNA. Reverse transcription is RNA to DNA. So you have to remember these points also. Now the next question we'll see. Deficiency of folic acid during pregnancy is strongly associated with which of the following outcome? Neural tube defect, cardiac anomalies, preterm delivery, placental abortion, gestational diabetes. You have to opt for the single best answer. So that happens to be the neural tube defect. So folic acid, other name is vitamin B9. It is required for DNA synthesis, repair, methylation, various steps around the DNA. So anything deficiency in the early pregnancy, high risk of neural tube defects such as spina bifida and anencephaly can be seen. Why not other things? Cardiac anomaly, preterm delivery, placental abruption, gestational diabetes. So they are not immediate effect of folic acid deficiency, which can be seen later. So we have to go for the closest answer, that is the neural tube defect, which is the best answer. Now, this is a quick overview of the various vitamins and their functions. Vitamin A, D, E, K, they are lipid-soluble vitamins. A is the retinol, beta-carotene, it's a proform of vitamin A. Deficiency will cause xerophthalmia, night blindness, keratinization of the skin. Vitamin D, cholecalciferol, this deficiency will lead to rickets or osteomalacia. Vitamin E, tocopherol, that is extremely rare deficiency, but it can cause it is seen, neurological dysfunctions can be seen. Vitamin K, it's also called as phyloquinones, menaquinones, various forms of the vitamin K are there. So it will lead, deficiency of it will lead to impaired blood clotting and hemorrhagic disease. Under the water soluble vitamins, if you see, it is all the water soluble vitamins and the vitamin C. So thiamine will B1 deficiency will cause beriberi, Wernicke coruscans. Vitamin B2 riboflavin deficiency will lead to lesions around the mouth, lips, and tongue, seborrheic dermatitis. Niacin deficiency will lead to pellagra. Vitamin B6, which is pyridoxine, pyridoxal, pyridoxamine, will lead to the pellagra-like situations. So B6 and B3, they can both come up with the pellagra situations because B3 is required for B6 formation. Then about the folic acid, we just saw neural tube defect and megaloblastic anemia. B12, also called as cobalamin, its deficiency will lead to pernicious anemia, megaloblastic anemic picture. Then pantothenic acid, burning foot syndrome. Then biotin, it will lead to impaired fat and carbohydrate metabolism and dermatitis. Ascorbic acid deficiency will lead to scurvy. So this image will help you a quick overview of all the water-soluble and lipid-soluble vitamins. So that's about the questions which appeared in 2024. Now we'll see about 2023, which other questions have appeared. So here is the question. Which of the following enzymes plays a key role in ammonia synthesis in kidney? So options: glutaminase, glutamate dehydrogenase, alanine transaminase, carbamoyl phosphate synthetase 1, and arginase. So the best option here is glutaminase. So what is the role of glutaminase? It's an enzyme which converts glutamine to glutamate, so which releases the ammonia in the process. It's a very crucial first step which happens in kidneys and in liver. So the ammonia from the inside the cell is trapped by glutamic acid, then it will form glutamine. So now when we want to detoxify that ammonia, that means the glutaminase enzyme is required. What about the second option, glutaminate dehydrogenase? It is required, but it is in a later phase which is required. So here they are asking about the kidney, and the first important step is the glutaminase enzyme. So that's why it gets the first choice. Whereas the ALT, it is involved in amino acid metabolism, but not for directly for ammonia production. And carbamoyl phosphate synthetase 1 and arginase, you can see these enzymes in the urea cycle, not in the per se in the ammonia production. So which of the following are the primary bile acid synthesized? Okay, so the options are deoxycholic acid and lithocholic acid, cholic acid, quinodeoxycholic acid, taurocholic acid, glycocholic acid, lithocholic acid, tauroquinodeoxycholic acid, glycoquinodeoxycholic acid, and deoxycholic acid. So they are asking, please note, it is a primary bile acids. So bile acids which are synthesized in the liver. So the option here is cholic acid and quinodeoxycholic acid. So cholic acid and quinodeoxycholic acid. What are the other options? So now we know primary bile acids which are synthesized from cholesterol are cholic and quinodeoxycholic acids. These acids will undergo conjugation either with taurine or glycine to form bile salts, that is the sodium or the potassium salts. Okay, the conjugated versions will combine with further with salts, that is the sodium or potassium, to further form, so which are water soluble, and they are required for fat soluble, that is the fat digestion. So from primary bile acids, we get secondary bile acids, which is happening in the intestine, and bacteria is required for conversion to secondary bile acids. Which are those secondary bile acids? Deoxycholic acid and lithocholic acid. Deoxycholic acid and lithocholic acid. So the options here which you had seen, okay, taurocholic acid, glycocholic acid, they are the conjugated versions. So they are not the primary bile acids. Lithocholic, taurocholic, again this gets ruled out. Tauro and glyco, so again the conjugated versions are there. Deoxy is again the secondary version. So the best answer here is the primary is the cholic acid and quinodeoxycholic acid. Next question. A 32-year-old Asian woman is diagnosed at 28 weeks of gestation with the gestational diabetes. Treatment with metformin is commenced. Which of the following is an insulin antagonist? Okay, so cortisol, free fatty acids, growth hormone, prolactin, somatostatin. So here they have asked insulin antagonist. So please remember, whenever you have a lengthy question, look for the last point. Look for the last point. What are they asking? Then start from the beginning. So they have asked which of the following is an insulin antagonist. So anything which makes sense over here, you have to figure it out. And then options. So option here is somatostatin. So this insulin release is inhibited by adrenaline or somatostatin. Okay. So insulin is synthesized as the peptide hormone. That is the prohormone. Then once it undergoes the proteolytic cleavage, the C-peptide is released, and then the active form is obtained. So this insulin is released in response to increased amino acids or free fatty acids and gastric hormones, which are released in response to eating, including the cholecystokinin, gastrin, or secretin secretion. Now, insulin release is inhibited by somatostatin. Now, from the options given, it is the somatostatin is the best option. Next, we will see which of the following nitrogenous base is unique to DNA and not found in RNA. So this is pretty easy. So the obvious choice, which is unique to DNA but not found in the RNA. So adenine, cytosine, thymine, guanine, uracil. Uracil is seen in RNA. So this is not the answer. Thymine is unique to the DNA. Rest of the things are seen in both DNA and RNA. The thymine is a pyrimidine base unique to DNA, and it pairs with the adenine through two hydrogen bonds. In RNA, thymine is replaced by uracil, which pairs with the adenine. So this distinction is essential for differentiating between nucleic acid, that is DNA, and RNA. So that was about the question. Next question we will see. Let us see. I had given you a tip that you have to look for the last segment of the lengthy question. Which of the following finding is most correct regarding diabetic ketoacidosis? So 23-year-old woman is admitted to hospital complaining of abdominal pain, vomiting. She is a type 1 diabetic, did not take her insulin today as she has been vomiting for 12 hours. On examination, she is tachycardic, feels cold and clammy. Arterial blood gas confirms diagnosis of ketoacidosis. Which of the following is the most correct regarding diabetic ketoacidosis? High blood levels of fatty acids, hypoventilation, increased blood volume, low levels of lactate or respiratory acidosis. So certainly whenever you see diabetes, it is not the respiratory, it is the metabolic version. And acidosis, they can be into anaerobic situations, so low levels might not be seen. And she is definitely having vomiting, blood volume will be low, but here it is saying increased blood volume. Then hyperventilation, she is tachycardic, she is having in ketoacidosis, there will be Kussmaul's hurried breathing will be there. So now in ketoacidosis, what will be the best thing? Here answer is high levels of fatty acids. So when insulin is not available in type 1 diabetes, so what happens? The lipolysis will happen. Lipolysis will release lot of fatty acids. Fatty acids further will break down. That is why beta oxidation release acetyl-CoA. Acetyl-CoA will be the substrates for ketogenesis. So here this will be the best answer. That is the high levels of fatty acids. Okay. So that's about so patients are usually severely dehydrated due to the state of osmotic diuresis and hyperglycemia. So this is about the case. Now we will see the next question. Extracellular calcium is bound to which of the following? Albumin, bicarbonate, calcitriol, fibrinogen, phosphate. So the answer given here is albumin. So now we need to know the calcium exists in three forms: bound form, ionized form, or complex form. So bound with what? Primarily with the albumin. So how much of the albumin is binding to the calcium? It is around 40 to 50% of the extracellular calcium is bound to the albumin. And then free, the rest of the next 50%, which is biologically active version, is the ionized form. Okay. Then a small percentage, around 5 to 10%, is complex with the phosphates or bicarbonate. So albumin is primary protein responsible for binding of calcium, influencing the total serum calcium levels. Hypoalbuminemia can reduce total calcium without affecting ionized calcium levels, which is physiologically active. So whenever you are evaluating, you encounter total calcium low, so look for whether albumin values are low or correct. So that will also guide you whether it's whether it requires a correction of the factor or not. Next question. Northern blotting is used for DNA, RNA sequencing, protein identification, antibody identification, or none of the above. So the answer here is northern blotting is required for RNA sequence. It is commonly employed to study the gene expression by analyzing the mRNA levels. That is the northern blotting. Now, what are the steps in northern blotting? First, they isolate the RNA from the sample. Then the RNAs are separated on a size using agarose gel electrophoresis. This will be transferred to the membrane, that is nylon or nitrocellulose membrane. Once that is done, that will be hybridized with a probe, a labeled probe complementary to DNA or RNA probe with specific targeted RNA sequences added. Okay. Then detection. The probe is detected using radioactive or chemiluminescent or fluorescent labels. The signals indicate presence and abundance of the target RNA. So these are the steps of northern blotting. Isolate, separate by electrophoresis, then transfer to the membrane, hybridization to the probe, and then detection using those radioactive probes. Now, similar another question which had occurred in the 2023 paper was southern blotting is used for what? So it is good to know that similar questions had occurred in the exam. So you will be very lucky in that case. So whether it is RNA, DNA, protein structure, identification of antibody, or none of the above. So southern blotting is for the DNA analysis. So separation of the DNA fragments by electrophoresis followed by probing and hybridization. So what are the steps here? Isolate the DNA from the sample. Use the restriction enzymes. Restriction enzymes will cut and cleave the DNA fragments, then separate the fragments. Okay, then DNA is denatured. Then it is transferred to the membrane, hybridized with the probe, and detection will happen. So you can see similar steps in this northern blotting also. Remember, southern blotting is for DNA evaluation, northern blotting is for the RNA evaluation. Next question, Wernicke's encephalopathy is most associated with the deficiency of which vitamin? Vitamin B6, B1, B2, B12, or vitamin C. So here the option is vitamin B1. So vitamin B1, it's also called as thiamine. It is a encephalopathy. Wernicke's encephalopathy is a neurological condition with a deficiency of B1, often seen in chronic alcoholics, malnutrition, or hyperemesis gravidarum in pregnancy. It is essential. This thiamine is very essentially needed for carbohydrate metabolism. Okay, so that is a point to be remembered. What are the other things which can be seen in vitamin B1 deficiency? Wet beriberi, dry beriberi, infantile beriberi. So these are some points around vitamin B1. Now we have a last question. What is the action of glucose 6-phosphate dehydrogenase? Whenever you see the word ending with ASC, that means they are asking about enzyme. So whether it is a production of ATP by aerobic metabolism, ATP by anaerobic metabolism, NADPH production in RBCs, production of ketone bodies, or none of the above. What is the action of glucose 6-phosphate dehydrogenase? So production of ATP by aerobic metabolism. Aerobic means you will immediately remember about glycolysis. We don't get this glucose 6-phosphate dehydrogenase. Anaerobic, we get lactate dehydrogenase. So here NADPH is the most appropriate correct answer. You can see this is the HMP pathway product. So some 10% of glucose 6-phosphate, that is glucose when the person is in a good well-fed state, glucose converted to glucose 6-phosphate by hexokinase, that is shunted to HMP shunt pathway. G6PD will help in production of NADPH as a cofactor, and the other so product formed from G6 glucose 6-phosphate is 6-phosphogluconate. Okay, so you have to remember this. So deficiency of this G6PD, the person can present with the hemolytic picture. So G6PD deficiency and anything which causes the production of the free radicals will cause the triggering of G6PD deficiency. That is about the some antimalarial drugs and all can cause that one. So these are the questions which had appeared in 2024 and 2023. So I hope you found this brief session on the topics of biochemistry was helpful. So stay in touch with the team for any further clarifications. So we will soon get back to you. Thank you so much. Take care. Bye-bye.